The theme of this conference is seizing the radioisotope opportunity from coast to coast. Yesterday we talked about what that opportunity is and where we need to go to get there, and today we go coast to coast. So I'm very happy to introduce our next session chair, who will be taking us for a regional theranostics showcase. I'd like to call up Lisa Taylor, who is the Director of Strategy and Business Development for Laurentis Energy Partners. Please welcome Lisa.
Good morning, everyone. Thanks to Scenic for giving me this opportunity to be your tour guide for today as we go from coast to coast. We're going to go on a journey starting in Vancouver, moving to Saskatchewan, arriving here in Toronto, and ending our tour in Montreal. So, with this regional focus, we really want to shine a spotlight on the innovation, the groundbreaking research, and the novel approaches to multidisciplinary care that are being done in each of these regions to advance theranostics. So, I want to introduce our first speaker from Vancouver, Dr. Joseph Lau. Dr. Lau is an assistant professor in the Department of Radiology at UBC. He's also a scientist in the Department of Integrative Oncology at the BC Research Institute. And he previously was an associate director at Alpha-9 Theranostics. And his research focus is on radiopharmaceutical development for cancer diagnostics and therapy. So welcome, Dr. Lau.
Good morning. Thank you for the kind introductions. I've been tasked to give an overview of our program in 15 minutes, so I'll try to stick to that timeline, but there's a lot to cover. So I figured that I start with explaining what we are as an organization. So BC Cancer, it's a publicly funded health organization whose main primary mandate is to deliver comprehensive cancer care to cancer patients living in the province. We have 6 regional centers located in Abbotsford, Kelowna, Prince George, Surrey, Vancouver, as well as Victoria. But in order to provide provincial coverage, we actually partner with about 31 community oncology network sites to better serve our patients. We also have 2 state-of-the-art research facilities. One is the Daly Research Center in Victoria. The other is the LJ Blackmark Vancouver Cancer Center in Vancouver. I've included in this slide are both our vision and mission statements. It really encapsulates the importance of the work we do on a daily basis. And when I was a graduate student, I really saw inspiration from this, and I hope it resonates with some of our staff and trainees that are present today. So it's easy to guess that most of the development of the Radiotheranostics Hub is centered around Vancouver. I'm part of the molecular imaging therapy program, and we're a coalition or a team of interdisciplinary scientists, nuclear medicine physicians, technologists, radio safety officers, medical physicists, as well as a lot of trainees. And together, we're trying to improve cancer diagnosis and treatments through the development and adoptions of novel radiotherapeutics, as well as innovative technologies. We collaborate in a growing network of national and international consortiums and programs, and throughout the talk, I hope you can see that theme emerge. In terms of research infrastructure, at Vancouver, we have a radiopharmaceutical facility that's overseen by Dr. Kuo-Shan Lin. It has a 19 MeV cyclotron, hot cells for both clinical and research use, shielded fume hoods, as well as a dedicated cold chemistry facility. The image on the slides, it's one of my favorite images of our buildings because it really shows how close our research center and our research center as well as the clinic is. So it's actually just separated 10 steps away. So on the bottom right of the picture, you see there's a garden pavilion. Right beneath that is actually our radiopharmaceutical facility. In BC, we have 4 publicly funded PET scanners, 2 in Vancouver, 1 in Victoria, as well as 1 in Kelowna. And just last month, we've installed the first long-axial field of view PET scanner, the Quadra, in Vancouver, and this was thanks to a funded CFI application. We do not have spec imaging capacity at our clinic. Rather, we actually rely on the Vancouver General Hospital to provide imaging for lutetium and actinium-based agents. So there's a little bit of inconvenience, but it also adds a degree of coordination by our staff. But it's only about like a 3 to 5 minute drive away from our Vancouver site to VGH. One of the things I want to mention is that Vancouver BCCA was designated as a comprehensive radiopharmaceutical therapy center of excellence by SNMI, and we're one of the 4 centers joined by University of Laval, UHN, as well as London Health. So one of our commitments is conducting rigorous clinical research, and I want to acknowledge at this time Nadine for helping put together some of these slides right now. So these are the ongoing clinical trials separated by types of radioisotopes. So we currently have 2 actinium-based trials, Excel and Pantha. Both target PSMA. They're sponsored respectively by Point and Eli Lilly for Excel. Pantha will be Bayer. So those are open for accrual, and I think we received and treated our first patients probably last month. In terms of lutetium, we have 3 agents that are currently being evaluated. We have HTK-03170, which is a radiotherapeutic developed in-house. Dr. Bernard had mentioned this agent from yesterday's talk. I'll probably review it a little bit later as I go through the preclinical development. But that's a phase 1/2 study. And at this point, I should also point out that Dr. Carlos Uribe will be presenting, giving an oral presentation on the preliminary results at the upcoming SNMI. We're part of the PR21 program, which is a pan-Canadian effort to evaluate the use of Pluvicto. As well, we are engaged with the NET Retreat program. So the study just recently opened, so we haven't had a patient yet. For the talk, we talked about actinium-based tracers, lutetium-based tracers. We also do companion diagnostic development. So 18F-BL40, it's a radiofluorinated compound, a PET tracer for imaging chemokine CXCR4 receptors. And so this was also developed in-house at BC Cancer. Again, it's a phase 1/2 study. Phase 1 component has been completed, and we're now waiting to initiate phase 2. What is coming up? There's the actinium dotatate trial, phase 1/2 study. This is sponsored by Ray's Bio. And what's interesting about this particular compound is, yes, these are the same somatostatin analogs that we've talked about, but we're now investigating it in a setting of breast cancer. There's many, many clinical trials that have been conducted over the years. I'm just putting up 2 of the more recent ones. We were involved in the SPLASH trial as well as PSMA4. So these were both phase 3 clinical trials, again, in context of prostate cancer. So what's going to happen? In 2026, we expect the full commissioning of the Institute of Advanced Medical Isotopes. This is a partnership between BC Cancer, UBC, as well as Triumph, and this is a full 5-story building with about 2,800 square meters of research and clinical use. We'll add 2 additional cyclotrons, as well as a series of wet labs. And so we've mentioned throughout the conference the need to have redundancy in manufacturing, and so this really addresses some of the potential shortcomings of single points of failures. Longer term, we expect BC Cancer, we're hoping to increase, and this is obviously contingent on funding, increasing PET CT scanning capacity throughout the province and expanding as well our radiotheranostics services. So all that is to be said is that we're obviously expecting novel isotopes and more clinical trials. So the radiopharmaceutical development at BCCA, I've listed, there's 5 primary principal investigators, Dr. Francois Bernard, Dr. Quoshan Lin, Armin Ramim, Carlos Uribe, and myself. And our research focuses are all listed here. But I want to say that we aren't alone. In BC, we have, and we benefit tremendously from strong collaborators at different research institutions at UBC, Simon Fraser University, as well as Triumph, whose expertise we tap in and leverage for isotope production, chelator development, as well as radiochemistry labeling techniques. So in the next 3 slides, I'll try to go through some select examples of targets that we've developed or focused on developing radiopharmaceuticals for. Some have been talked about by Dr. Bernard, but I'll try to quickly go through each of them. So the first one is the somatostatin receptor, where we leveraged, again, a one-step radiofluorination strategy to develop an imaging agent for neuroendocrine tumor. And what's more important to us, besides the fact this is the first proof of concept, was that it's actually the first radiopharmaceutical developed in-house of BC Cancer that we translated. So the imaging study has been concluded. We show good tumor uptake, high image acuity, and we believe with the superior imaging qualities of fluorine-18 over gallium-68 that this will lead to better adoption and increased accessibility. Even though the half-life is not that much difference, 68 minutes and 110 minutes, it is still a meaningful amount. You can actually get a lot more patient doses using a fluorinated compound. With PSMA in the middle, I don't think we need to introduce this target too much. All that said, the HTK-03170 compound is a modified peptidomimetic, so we've modified the traditional lysine-durated glutamate construct, and we've also conjugated onto it, or engrafted onto it, an albumin binder derivative to modulate the PK. Collectively, these changes allow us to increase the tumor uptake and retention of these compounds by about, and dose delivery by about 10-fold compared to Pluvicto in preclinical models. So this study, again, is in phase 1 and phase 2 evaluations. On the far right-hand side, we have CXCR4. Dr. Bernard gave me a lot of credit for starting this program, but I would like to pass the credit to our graduate student, or our current now MD PhD, former MD PhD student who's at University of Ottawa right now doing his residency, Daniel Kwan. And he really took this project steps above what I originally envisioned. But we took a cyclic inhibitor, peptide inhibitor against CXCR4, made modifications, and now we have an agent that performs, at least in preclinical models, much better than gallium-68 pentaxifer, which are on the left-hand side. 3 other targets here, melanocortin-1 receptor, so this is overexpressed in malignant melanomas. Problems with this system is the fact that these peptides are readily cleaved by peptidases existing in the body. So our former staff scientist, Dr. Chang-Cheng Zhang, made selective modifications to this compound, doing tedious amount of chemistry work to kind of optimize this to make it more stable. In addition, working with the Triumph group, with new chelates, they were able to make a compound derivative that was enabled visualization of a receptor expression about 1,000 copies per cell in this tumor model. So this is very important work. Middle, we heard about gastrin-releasing peptide receptor in this talk yesterday. And this receptor is a G protein coupled receptor that's expressed in both prostate and breast cancers. In the prostate setting, it's hypothesized that this can serve as a complementary role to PSMA negative cancers. And we've developed a series of linear peptides with modified pseudopeptide bonds that, again, reduce not only pancreatic uptake, but also increases stability and tumor uptake of these constructs. On the far right-hand side, fibroblast activation protein. So if anyone remembers, there was a seminal paper that was published back in 2019 by our colleagues in Heidelberg, talking about the fact that FAPI could visualize about 28 different types of cancer. So this is potentially a pan-tumor or tumor-agnostic agent. And so Dr. Kuo-Shan Lin and his student, Shreya, actually played a lot with these sort of compounds, small molecules, modified them, and were able to increase tumor uptake and retention. And I'm proud to announce that we're going to be bringing this agent to clinic over the next years. Yesterday, I think Dr. Allman from Telix talked about CA9, and particularly the fact that colorectal cancer was a passion of hers. CA9 is a passion of mine because that was my PhD project. So I have a little bit of alignment there. This is a target overexpressed in clear cell renal cell carcinomas, about 80 to over 90%. And this is due to the fact that there's upstream mutations in the tumor suppressor gene, which is von Hippel-Lindau, that causes the constitutive upregulation of this protein. So what I'm showing here is actually just zirconium-89 labeled radioimmunoconjugates, and this was done in collaboration with the National Research Council of Canada. And really just to showcase that not only do we work with peptides, some small molecules, we have the ability at BC Cancer to work with larger proteins as well. In the middle, so this is kisspeptin, some of the work that's developed in my lab, probably considered, I think, a new target in the field, considering the fact that its protein was actually only identified back in 1996. A little fun fact, the kisspeptin gene was isolated or discovered in the town of Hershey, Pennsylvania. First Hershey Kisses, kisspeptin. So it's expressed, traditionally this role has been investigated as an anti-metastatic properties in melanoma. But more recently, there's been growing literature supporting the fact that this could be an oncogene, especially in the context of triple-negative breast cancers. So we were fortunate enough to look at this target, develop peptides, and what I'm showing there are really first-in-class radiopharmaceuticals that achieve really good uptake in an overexpression model. Last but not least, I want to give some credit to and acknowledge some of the work from my grad student, Simran, who's been working tirelessly for the neurotensin receptor. So this is a G protein-coupled receptor that's overexpressed in solid malignancies, mainly pancreatic cancer as well as prostate cancer. So we're looking at both peptides and hybrid approaches to actually improve stability of these agents, and she's making good progress. Instead of listing out all the targets that Francois and Quotient are all working on, I just decided that maybe highlight some of the targets that I'm also pursuing in my lab. These include other G protein coupler receptors, glycoproteins, and receptor tyrosine kinases. I mentioned maybe 5 slides earlier that we engage in national and international collaborations, and so I would really like to take this opportunity to highlight a grant that co-led by Dr. Francois Bernard and Katerina Remgada. And we received a $23.8 million grant over 6 years in 2023. And it's a pan-Canadian initiative. So there's 18 principal investigators involving 8 academic institutions. The title of our talk is The Rare Isotopes to Transform Cancer Therapy. I've listed the aims of the grants or paraphrased them here. I'm not going to go through all of them in the interest of time, but I would like to highlight 2 features. The fact that we have built in, well, the box is a little off, but we've built in funding to enable first-in-human studies for both diagnostic and therapeutic agents. And as I mentioned earlier, the FAPI agent that we would like to bring into clinic is supported by this initiative. And also, throughout the course of the conference, we've heard about the need of training and the next generation scientists. As of before I flew into Toronto, I emailed our coordinator for the program and asked how many trainees are actually involved in this program. And the latest count is 77 trainees. So we're 77 trainees in 18 labs that are working in this initiative, and we expect that number to grow. And it's something that we're very proud of. I'd be remiss in the last couple of minutes to not only talk about industry and academia. I thought I would, with the number of industry participants here, there's also a growing commercialization space in BC beyond just radiopharmaceuticals. But related to radiopharmaceuticals, we have Artemis, Alpha 9 Oncology, Abdera, as well as Acinta. One commonality, they all start with A, but 3 out of 4 of them actually are spin-off companies from either Triumph, UBC, or BC Cancer. And a quick congratulations for the Artemis group. They were acquired by Telix in 2024. So to summarize, between all the research institutions in BC, we have a really integrated ecosystem that enables the full spectrum of radiopharmaceutical development, from isotope production all the way to clinical trials. We have strong research infrastructure to support innovative research, and there's more forthcoming. We're always patient-centric. We focus on the delivery of patient care, and we're trying to move new agents beyond PSMA and even beyond lutetium and actinium into future clinical studies. Last but not least, at BC Cancer, we really value collaborations. We cannot do this alone. It's not a siloed mentality. We appreciate and understand that academic, clinical, as well as industrial partnerships are really what's needed to drive patient care. So with that, just want to acknowledge everyone in the molecular imaging therapy program, both past and current lab members, as well as everyone on the committee for organizing this meeting. So happy to take any questions at this time. Thank you.
I have a question for you, if no one else has got one. I realize this is kind of like asking you to name your favorite child, but if you had to choose a favorite target in the research that you're doing at your institution, do you have one?
Francois and Coach, I might not like this, but I like the Kisspeptin project since it's my first project independently as a new researcher, so it's my second year as assistant professor. But I think all of them were really unequivocally interested and committed to all of them. I think FAPI has a lot of potential, and the first one in clinic, so we're always excited about that. Thank you.
It's a very safe answer.
All right, thank you.
All right, so now we're going to go from Vancouver to Saskatchewan, where we're going to hear from Dr. Kate Dadakova about Saskatoon's innovative use of targeted radionuclide therapy for infectious and autoimmune diseases. So a little bit about Kate. She joined the University of Saskatchewan as the Fedoruk Center of Nuclear Innovation Chair in Radiopharmacy. That's a bit of a mouthful. And she is a professor of pharmacy. Prior to that, she held a professorship at the Albert Einstein College of Medicine in New York, where she was a professor in radiology, microbiology, and immunology. And something that stood out in your profile is that she was one of the top 2% of cited scientists worldwide, according to Stanford University in 2021. So I feel like you might be too busy for my Netflix show recommendations. So why don't you come up on stage and tell us about what's going on in Saskatoon.
Good morning, everybody. So first of all, I would like to thank the organizers, especially Amanda Boyle and Melody Greaves for inviting me here to speak. So I will be talking today about an area which I think wasn't discussed a lot here, but though, again, Dr. Boyle discussed yesterday the imaging of neuroinflammation. So I will be talking about multiple sclerosis a little bit in my talk. So let me see if I can. All right. So you already heard about targeted radionuclide therapy through the whole day, yesterday and today. So radioimmunotherapy is a subset. So basically the targeting vehicle is an antibody. And radioimmunotherapy has certain advantages when we're talking about traditional applications in cancer and also in other applications, that it does not depend on a lot of multi-drug resistant mechanisms. So basically, it is difficult, say, for cancer cells to pump out the antibody once it binds to this receptor. And also radioimmunotherapy is not dependent, at least a lot, on the immune status of the patient. And this is extremely important when, again, one is talking about applying radioimmunotherapy to other areas than oncology. And labeling of antibodies is pretty straightforward. That kind of technology has been developed decades ago, before we joined the effort. And here on the slide, you see that we're attaching different linkers, for example, DOTA, which is a macrocycle, to the antibody via amine group, and then labeling it with a variety of radionuclides. Here it's one example from our paper in oncoimmunology, because we also work as everybody else in cancer. And the isotope there was actinium-225. At least these were the days when we could still afford actinium-225 as an academic lab. So this is the list of radionuclides which could be used for radioimmunotherapy. This is not by far an exhaustive list, but as you can see, both alpha and beta emitters are represented there, and the half-life of those alpha and beta emitters vary a lot, from 46 minutes for bismuth-213, which is a daughter of actinium-225, to actinium-225 itself, which has almost 10 days half-life. And in our experience, and in our other labs' experience, antibodies are good as gold for delivering any of these radionuclides, even 46 minutes half-life bismuth-213, which has been demonstrated times and again, both in the lab and in the clinic. Again, it's easy to label the antibodies. That's why we think it's a very versatile approach, radioimmunotherapy, and it could be applied to other areas. So why use radioimmunotherapy for oncological applications? So here are some numbers, and these are the numbers from Canada alone. We are not the largest country in the world, as you know. So from cancer, we lost 86,700 people. Most recently. Simultaneously, 5,500 people died from infections. This excludes COVID, and that actually excludes a lot of opportunistic infections, infection of people who die in intensive care units, because they will be listed like died of cancer, died of heart disease, complications from those, right? But in fact, they died of many of opportunistic infections. And more than 42,000 people died from autoimmune diseases. So do those people matter? I think they do. And I think we should make an effort to use our tools, which are amazing, to try to help those patients. So first I will talk about radioimmunotherapy of multiple sclerosis, and the second part of my talk will be about radioimmunotherapy of infectious diseases. So multiple sclerosis is a neurodegenerative disease when there is a cohort of T cells and B cells, and especially T cells, which start to affect the protective sheath of the nerves, so-called myelin, and it causes all kinds of horrible neurological symptoms. People have flares. It is really debilitating conditions. And for some reason, Canada has one of the highest incidence of multiple sclerosis in the world, and Saskatchewan is actually the champion in Canada in a sad term in having the highest incidence of multiple sclerosis in Canada. So it has been established, and you see the schematic on the slide, that PD-1 receptor, programmed death protein 1, which is one of the major targets in immuno-oncology in cancer, is for some reasons, and the reasons are on that cartoon, but I'm not going into immunology for the sake of time, is expressed by that subset of aggressive T cells, which are actually responsible for attacking that myelin on the nerves and causing all those symptoms in multiple sclerosis. So we hypothesize that if we can target PD-1 with radioimmunotherapy using the same antibodies which are already on the market for treating of cancer, we could eliminate that subset of aggressive T cells, and by doing so, alleviate the symptoms of multiple sclerosis. So what we used the same model which Dr. Boyle yesterday described. This is experimental autoimmune encephalomyelitis. It's a mouthful, so we call it EAE. And what you could see here, first we imaged our mice with EAE who develop the variety of neurological symptoms, from hind leg paralysis to death. It's like the highest clinical score of 5. And we took the antibody, anti-PD-1 antibody, labeled it with zirconium-89, and lead-203 to perform PET and SPECT respectively, and we looked at the lymph nodes. As you can see on the left, at least it's my left, right? So where there are EAA-induced mice, there is a lot of uptake of the antibody in the lymph nodes. This is where those malicious T cells are hiding, in those lymph nodes. And in healthy mice, there is no uptake in those areas where the lymph nodes are located. The same applies to the brain. So that was really encouraging to us, and we decided to move into therapy. So here you see the clinical score of our mice treated with lutetium-177 labeled anti-PD-1 antibody, the same antibody we used for imaging of mice with EAA. And the red line shows the lowest clinical score of the mice treated with that specific antibody in comparison with unlabeled antibody, non-specific antibody control. So basically to show that the effect is a PD-1 specific, not just untargeted radiation specific, and also, of course, non-treated controls. So these were really encouraging results, and then we proceeded, which were published last year in Journal of Immunology. So our graduate student who defended this week, defended his PhD, now he's going to medical school, Connor Frank, the first author of that paper. So he also combined radioimmunotherapy with a drug which is approved for treatment of multiple sclerosis, so-called fingolimod. And he showed that combination therapy is actually much more effective even in preventing the flares of multiple sclerosis. So this paper just got accepted in Frontiers in Medicine, and we hope that this approach will go somewhere. But also, conclusion for this part is that this approach targeting T cells, right? And T cells are implicated in many other really serious high numbers autoimmune diseases, such as diabetes, for example, or rheumatoid arthritis, systemic lupus erythematosus. So this approach of targeting T cells with radioimmunotherapy, I think, has potential in other autoimmune diseases. So now I will be moving towards the second part of my talk, which is radioimmunotherapy of infectious diseases. So why do we need to apply radioimmunotherapy to infectious diseases? Of course, I'm not talking about some applications such as common cold or things like that. These are much more serious infectious diseases I'm talking about. And first, of course, is that there are a lot of so-called opportunistic infections which affect people with suppressed immune system. And medical progress created a lot of such individuals. Say, if somebody is a cancer patient who went through several or sometimes even just one round of whatever cancer treatment they're being prescribed, chemo, immuno, or external beam radiation, they become immunosuppressed. They are organ transplant patients who are suppressed for the rest of their lives in order not to reject the organ transplant. The HIV-infected individuals. Nowadays, there are people who lived through COVID, and because they were treated with high doses of corticosteroids to suppress the inflammation, they also became immunosuppressed, and the list goes on. And of course, there are a lot of resistant pathogens, especially bacteria, which are resistant even to antibiotics of last resort, such as vancomycin. And there are also microorganisms for which there are no treatments. So back when I just started as an assistant professor, and that was a long time ago, in the early 2000s in New York at the Albert Einstein College of Medicine, that was the first paper which we published in PNS, where we infected mice with Cryptococcus neoformans. Cryptococcus neoformans is one of the major fungal opportunistic pathogens. It used to be a real threat for people with HIV before the introduction of antiretrovirals in 1996, but still a lot of patients with suppressed immune system, especially in developing worlds. Actually, the numbers are hundreds of thousands of people die from cryptococcus, primarily fungal meningitis, every year. And this slide shows you that when we treated mice with organism-specific antibody labeled either with beta emitter, rhenium-188 we used at that time, or alpha emitter bismuth 213, we prolonged the survival of significant number of mice. We actually just had to finish the experiment somewhere, right? And that's why it was cut at day 75. We killed the fungal cells directly, as those colony-forming units show. And the image below shows you where the antibody is. Back then, it was 2003, we didn't have micropet or microspect, so all we had was a handheld camera for thyroid imaging, which they use in patients. So you see here the upper part of a mouse body, the red area are the lungs, that's where the fungus congregates, and the second area is the brain, because all those opportunistic infections, they have the ability to enter the brain very early via the Trojan horse approach, and, but antibody can chase them there because the blood-brain barrier becomes compromised. And then we continued working, so in 2010, this approach even ended on the cover of Journal of Infectious Diseases. That's the Journal of Infectious Disease Society of North America, where we compared radioimmunotherapy, just one shot, with the standard of care, amphotericin B. That's what they prescribe to people with invasive fungal infection. And as you can see, we were able to almost completely suppress the infection in the lungs and the brains. This is the logarithmic scale you see on the graph, in comparison with the several days administration of amphotericin B, which is, by the way, horrible for the liver, and still couldn't do anything with the infection. So later on, we realized that it's actually much more useful to find one specific pan-antigen, which is expressed by any fungal pathogen, which is out there, develop one antibody against it, and use it for any case. So we use it first in blastomyces dermatitis. The antibody was against beta-glucans. And on the left, you see the results published in 2020 when we labeled the antibody with bismuth 213. This, I think, was the last year when we could afford to buy actinium bismuth generators before they become like obscenely expensive. And we were able to eliminate the fungal burden from the mouse lungs. And then we tested the safety of this approach in dogs. Nothing bad happened with these dogs. They just sort of were watched for 6 months, and then they went on to do what they're always doing in veterinary college, which we have, and then they, like in a couple of years after being that veterinary dog, they get adopted by students or faculty members. So we showed that that approach is completely safe. Then we also expanded it. Our sort of last foray into this area was in 2023 when we, in a mouse model of mucormycosis, which is a horrible disease, emerged after COVID. Here you see the image of the patient. This is Lancet. This is not our data. This is like from their clinic, showing those fungal lesions inside and out of the patient. Again, these are the people pretreated with corticosteroids, completely immunosuppressed, and those opportunistic fungal pathogens, such as the one causing mucormycosis, are coming really seriously on them. And our paper showed that we could do it in immunocompromised mice, which sort of resemble the immunocompromised patients with lutetium-labeled antibody, the same we used before, which binds to beta-glucan. And the last vignette is what we're doing currently. Right now, the effort is going on. This is infection of prosthetic joints. We know that population is getting older, but it's not applicable only to older people. Everybody nowadays seems to have like replacement, knee replacement, hip replacement, shoulder replacement, whatever. At least 2% of those getting infected, and they're not infected because the surgeons do not wash their hands or ours are dirty. This is because the infections comes from patients' own microbiome, and some patients' microbiomes are more aggressive than others. What happens then, you see that those infections form biofilms on prosthetic joints, and no matter how high dose of antibiotic you will give to patients, biofilms do not respond. And amazingly, the mortality, like that graph from Lange et al. On the left, shows you that 5-year mortality from those infections is actually higher than mortality from melanoma, prostate cancer, and other types of cancer, and breast cancer. So just puts things in perspective. So we collaborate on this with researchers from UT, University Medical Center Utrecht in Holland. They developed an antibody which binds to bacterial cell wall. First, we did experiments in vitro showing that you can actually destroy the biofilms in vitro with antibody radio-labeled either with actinium, bismuth, or lutetium. Then it was shown in a mouse model where the infection was just in the soft tissue. But what is being done in the lab right now, we have a resident, orthopedic surgery resident from Holland in the lab. He put those titanium rods in mouse bones, and we treated them with antibody labeled with lutetium-177. You see that there are really good accumulation in the infected joint in the rats on the top, and nothing happens when you just inject lutetium DTPA. We were able to lower the infection burden a lot with just one injection, and now we're combining the treatment with antibiotics, and again, it shows much better results. So conclusions are that radioimmunotherapy is a universal and versatile therapeutic modality which could be adapted to the needs of patients and medical community, and biological half-lives of the antibodies actually provide really good match with currently available radionuclides such as lutetium, actinium, and such. And I would like to thank my lab. These are all amazing, really dedicated, bright, wonderful people, and a lot of collaborators, and of course the funding. We wouldn't be able to do anything without funding. Thank you so much.
Anyone have questions?
No?
I have a question.
Okay, sure.
So thank you for that very interesting discussion, and as
Yeah.
someone that has a family member touched by MS, really interesting to see the research. You have a room full of people that are very vested in the radiotherapeutic space. Do you have any resource challenges, needs, things that we can help you with to sort of advance things?
Well, I think this area sort of still lacks the interest from industry, unlike cancer. So I think there is maybe under-realization of the seriousness of problems which patients in other than oncology areas are facing. And as I was trying to explain at the beginning of my talk, that a lot of oncology patients actually cross into that infectious space at a certain stage of their disease, and that's what they die from, which is considered like a complication from treatment, but these are infections which could be probably treated. And again, sort of, I always talk at all meetings recently that the way the price of, say, alpha-emitting isotopes is going right now, the research communities in the universities, no matter how many federal grants I have, I cannot afford to buy actinium anymore, honestly. I mean, we're getting the quotes from different suppliers, and they're just like getting higher exponentially. So I think the producers need to direct at least a little bit towards the academic researchers, otherwise like research will wither away in alpha-emitter space. So that's our challenges.
Very good point, and I think a good message for this room. Thank you so much.
Okay, thank you.
All right, so now we're going to hear from Dr. Luke Brzezowski on the roadmap that they have here at UHN for the Theranostics Center that they're building. And I wanted to First, to tell you a bit about Luke. He is the Executive Director of the Theranostics and Core Facilities at UHN. He has also served as President of the Board for CanProbe, a startup specializing in molecular imaging and theranostics. And Luke has a doctorate in nanotechnology and nonlinear optics from the University of Toronto and was awarded the prestigious 2003 Governor General's Gold Medal Award for his research excellence. So, why don't you join me up on stage, Luke?
Thank you, Lisa. So I think for the rest of this session, we'll switch gears and we'll go more operational. So I will tell you the story of how we have been preparing for a large-scale theranostic standard of care at UHN and how we have been building from research experience. So first, let us anchor ourselves. Why are we doing all of this? So here is the ethical motivator. On the left, you see a patient who is suffering from cancer, loss of black dots. That's bad. 4 injections of theranostics, they're gone. So it's a wonderful thing. We've given this patient, we collectively as humanity in the clinical space, we've given this patient additional months or years of life. Here is additional motivator. Over the next 10 years or over a decade, we are thinking or we are projecting the market will grow by a factor of 4 for all of the radiopharmaceuticals, and actually the therapeutics themselves will grow by a factor of 10. So we have to prepare ourselves for it. So we have the ethical motivator, that was the previous slide, and this is the business, the operational motivator. And presently there are actually 67 assets in clinical development. We have 2 on the market right now. And these 67 assets are for 25 unique targets across 20 different tumor types. So it's really booming. There is more in the preclinical space. And it's an effect of merging decades of nuclear medicine innovation with cutting-edge biotechnology to target cancers with unprecedented specificity. So it's really happening, and we have to prepare. So we have high aspirations at UHN. We want to have a theranostic center. We want to really lead the world. We want to have world-class talent, retain and attract, train and educate. We want to have discovery research. This field is really uniting biology, chemistry, physics. It actually uses words like radiobiology, biochemistry. It's always crossing the disciplines in medical physics. We really care about strategic partnerships, and it's with industry. It's in clinical trials, but also in development. We work with government. We talk to them frequently, as well as we work with academia in clinical trials and other things. Clinical trials, we work on sponsored, on industry sponsored, as well as on investigator initiated. We need to have good infrastructure for discovery, but also for patient care. And all of it is always with an eye on seamless patient care. They have to have access to good standard of care, as well as to access to the most innovative clinical trials compounds. Where are we doing it? At UHN. UHN is a small town. It's 40,000 personnel. Everyone is involved in research. We are all dedicated to translation. We care for about 1/3 of a million patients. It's about 3 billion revenue, and over 600 million of this is going into research. So a good place to be doing this. About half of what we do is in cancer, and cancer, as in most of the places, is standing on the 3 legs of chemotherapy, radiation, and surgery. And all of these disciplines, and definitely the first 2, are very important to bring in theranostics as standard of care. So into this environment, we are dropping theranostics. And as with any new therapy in cancer, we have 2 main goals. Prolong the lives of people facing cancer, and then together with our partners, provide state-of-the-art standard of care and innovative theranostics ecosystem. So how we got here, what is it that we are doing, how we have been preparing ourselves. Nuclear medicine and then theranostics have been a priority for UHN for over a decade. Working with our partners from Hamilton, from CPDC, we've created CanProbe, which was a really pioneering thing where we worked on new compounds for nuclear medicine. It was diagnostics at this point. We built a cyclotron facility, we've commissioned it, we started supplying trials, we started supplying more and more, thousands of doses a year. We've really been the ones that have introduced it to our community, as well as ensured together with our partners that this is something which is really working in Ontario. We then moved a bit into therapeutics. We had some commercialization successes. We even had a drug establishment license, and we've reached 2,200 doses, in diagnostic doses a year as our maximum volume. And then actually we've pivoted, and over the last couple years we focused more on innovation. And here's a bit of a story. So you don't have to, of course, go through all of it, but if you focus on the two top rows as well on the bottom one, this tells how we've been pioneering the diagnostic dose production and delivery across Ontario. So we deliver thousands of doses, and the two that we focused on are the diagnostics, which are for PSMA prostate cancer and for neuroendocrine tumors for Lutathera. So with these two compounds, we've established a culture in Ontario, which then permits to have diagnostics for the two standard of care therapeutics, Lutathera and Pluvicto. You can also see the volumes going down over the last couple of years, and that's a good thing. There are more suppliers, things are being done closer to patients. We are again pivoting our facility more towards innovation. So now therapeutics, that was diagnostics. Last year, we've done about 400 injections and only about 100 of the new radioligand therapy at UHN. So that was Lutathera as well as clinical trials. Extrapolating what is happening around the world, we think that it will grow. It will grow by an almost order of magnitude over the next several years. The main driver will be Pluvicto, which has been recently approved and then approved for reimbursement in Ontario. And we expect that this one alone will increase our volumes to over 2,000 over the next 5 years. So it's a really big challenge. We have to, again, prepare ourselves, and as always, everything that we do at UHN is for ourselves, our patients, and for the healthier world. How we do it? We want to establish the Silver Family Theranostics Center, so infrastructure that we can handle it safely. We want to be able to deliver it safely as a standard of care, so operational procedures at the much larger volumes that we've done so far. We want to retain and attract leading innovators and trainees. We want to keep modernizing our innovation pipeline, so always do good research. Deep partnerships with pharma, we are very much open to it, as well as have open dialogue with government, and it's been very good and successful so far. It's not easy. There are challenges, logistical, supply chain, how to deliver it. There's the half-life, which is always chasing us. This is what makes it so distinct from any of the other disciplines. And you have to keep explaining it to hospital, to government, that half-life is something which says where you have to make it, how you have to deliver it, and time is money, very much so here as well. We need to ensure that we can get proper reimbursement for all the proper pieces. And again, it's not just a pill in a box on a shelf, it's decaying, it's very, very expensive. Each dose can be over $30,000. You have to ensure you get reimbursed, that you handle it, and that you get reimbursed for hospital costs. Workflows are very disciplinary. We all have to work together in it, as well as keep training and educating. A bit more on how multidisciplinary it is. We really work across all the departments. Nuclear medicine is the place where the theranostics are administered. Radiation oncology and medical oncology prescribe it. All these physicians work together at tumor boards and multidisciplinary cancer conferences. Medical physics helps with dosimetry. Laboratory research always teaches us new things. Translational research permits us to translate them to standard of care, and medical imaging is required for diagnosis, triaging, and follow-up. When we were preparing ourselves for large-scale standard of care, we talked about 20 different hospitals from around the world. And we talked about what you do, how you do it, what is safe, do you do dosimetry, who is doing what, what's your governance, who do you buy it from, how much does it cost, how do you handle it, extremely complex things. We've learned a lot. And we have at UHN been kind of on the bottom line as to when you look at what we can handle and how much we do. With recent approval of Pluvicto, we are already doing over 5 injections a week, and this is growing, so we are moving closer to the center of the pack. And with investment into infrastructure, we'll be open for several thousands or many more of injections a year. So we are really preparing ourselves for this extraordinary growth. When waiting for approval of Pluvicto for reimbursement, we also wanted to focus on 2 things. Be able we can provide safe standard of care, as well as be able that we can afford it. So we have multidisciplinary groups which were working on these 2 things. And again, they took people from nuclear medicine, oncology, medical physics, safety, research, everyone that had to be involved. And we mapped it out. So we mapped out that depending on volumes, what we need to do at which stage, and this is our roadmap. Depending on what numbers we will hit, what can be the projections, what government is telling us that they will be able to provide. We know when we need to hire additional people, when we need additional equipment across all the disciplines. Of course, we'll have to adjust to reality, but we know quite a lot as to what is expected. When it comes to finances, you can chop it into 4 different places. Starting from the end, there's capital. Of course, we get some provincial funding. It's not enough for the growth we expect and for the research we want to do, so we are fundraising for it. Physicians have to be also reimbursed properly for everything from follow-up to analyzing images to approving dosimetry plans. Radiopharmaceutical costs is not really what hospital is involved that much in, but we are, of course, interested that there is agreement between pharmaceutical companies and the government. And very important, hospital costs. It's expensive, it's multidisciplinary. So we struck a working group when we did very precise case costing, looking at every step from taking the patient in, administering, following up, imaging. And we've submitted this case costing to the government, to Ontario Health. We did it together with other partners from Ontario. And at the same time, we were talking to the government about the importance of theranostics for patient care, why we need it, what it is, educating them, listening to them, very open dialogue. So we were very disappointed when the announcement was made in November last year that Pluvicto will unlikely be funded because PCPA could not reach an agreement with Novartis on the proper pricing. But very quickly, we were very happy because Ontario decided to fund it regardless. And because of this, other provinces will likely also follow suit. PCPA has even reversed its opinion. 3 other provinces right now, in addition to Ontario, are also getting Pluvicto reimbursed, and we are administering already it to patients. We had a room ready for it, and the reason why we were able to have this room is because we do a lot of research. So this is the room that we renovated actually for PSMA, lutetium-177 therapy. So when Pluvicto was approved for reimbursement, we had the room ready, we had people trained, we had safety procedures, we knew who is doing what, how to receive it, where to inject it. So again, lots and lots of learning from research, and when new compounds will be coming on, we'll be building on our research experience. Pretty much all we had to do to make this room ready for standard of care was apply to Canadian Nuclear Safety Commission. Theranostics have grown quite organically at UHN, and as a result, there are lots of band-aid solutions. Some things are done across the hospitals. Imaging is done here, injection is done there, patient is waiting somewhere else, physicians are in another office, people have to run with radioactive material from building to building. It's far from ideal. It was really for very low volumes. So we are building this. We secure the space, and we are building a professional theranostics center at UHN. It will have 2 PETs, 1 SPECT. It will have 6 injection bays, 2 therapy rooms, which can be further divided into additional injection bays. So overall, up to 10 injection bays with a hot lab. We believe that this will give us a seamless flow. It will make patient journey much easier. It will be a big return on investment for efficiency, that our technologists will not have to walk in between the buildings. And we are in the midst of fundraising for it with the Princess Margaret Cancer Foundation. Our approved goal is $30 million. We have 15.5 committed today. Priority is to get the infrastructure. We don't want to be house poor, so we want to be ensured that we can build it in time. Clock is ticking. And big thanks to Alan Silber family and friends for a $10 million foundational gift. A bit more on research and innovation. I keep, or I at least try to emphasize that we were able to prepare ourselves for standard of care and do it safely and smoothly is because we work closely together across departments and learn from research. So we have a theranostic innovation pipeline. We can discover new theranostics. We can radio label them in-house. Again, people are learning how to handle it. We can do preclinical testing. We can do large batch production. We can deliver it across the hospital. We have imaging equipment to test it, and then we can analyze it. Many of the people which are right now applying standard of care have been part of building it, contributing it, keep using it, and that's how we can translate these learnings to standard of care. We really also like clinical trials, and we like it for several reasons. We believe in research, we believe in innovation. We want to know what's coming. It's quite a good business as well. But most importantly, it gives our patients access to the most innovative therapies. To date, we've had only 23 trials which are ongoing, opening, and evaluated across all the different sites across human body for many different targets, for many different radioisotopes, and for all the phases 1, 2, and 3. We work with quite a lot of companies, so there is a lot of interaction there, and it's knowing what's in their pipeline, it's thinking how we will implement it, it's understanding what they want, it's providing them clinical input. One of the things that we constantly keep hearing from our partners is that things have to happen faster. The patience of radiopharmaceutical companies when it comes to having clinical trials has a very short half-life. So we have an institutional priority of doing it fast. It's called IA45, and that means that within 45 days since the submission of an application, we want to have approval to start a clinical trial. We are actually successful to push several trials through it, and it's moving quite well. We'll see when we can implement it fully, but this is something that we are determined to do. Additionally, we've worked with other hospitals across the province to come and pharma to come up with Ontario Universal Agreement for Clinical Trials, and that's to shorten the time for negotiation of contracts. So those are non-negotiable terms to which lots of pharma agrees, and this should really, and it already has been successful, shorten the period of time. So we've been doing standard of care of Pluvicto for about half a year. Maybe next year I'll tell you how was the first year of implementation. I know that London has a section about it. It's good to be prepared. There are lots of surprises, but it was good to prepare and lots of learnings that I'm sure our colleagues from London will share with you. So here is where we are going to close with the same slide: world-class talent, discovery research, strategic partnerships, clinical trials, infrastructure, and all of it for seamless patient journey. Thank you.
We have time for a question if anyone has one. I have a question.
Okay.
So thank you for that presentation. I'm really excited about the Theranostics Center that you're going to be creating. I think along with many in the room was really pleased when funding was put in place in Ontario for Pluvicto. From the practical experience that your center has had so far, sort of transitioning from trial patients to ongoing clinical patients, are there any sort of lessons that you could share with those centers that are just sort of starting up their programs?
So I would say that talk to the people who have been running your clinical trials, map out the process, decide what is specific to research, what is not. It's a really lot of very good learning experience. Lots of the roles are the same. Technologists will do the same thing, physicians will do the same thing, but at the same time think about what you can approve, because it's not a one-off, but it is large volume, so a lot of the roles can be combined and some of the things can be simplified. We are going through these growing pains as we speak.
Thank you so much.
Thank you.
So the last stop on our regional road trip is in Montreal, where Dr. Daniel Junot is going to talk to us about radiotheranostics at the forefront, showcasing Montreal's pioneering expertise. So first, a bit about Dr. Junot. He is the head of nuclear medicine at the Centre Hospitalier de l'Université de Montréal, or CHUM for short. He's also the medical director of the cyclotron and radiochemistry unit at the CHUM Research Center. And his research interests are focused on radioligand therapy and new theranostic approaches, including phase 1 and phase 2 studies of new theranostic molecules. So please come up and let us learn more.
All right, good morning, everyone. It's a pleasure. It's a pleasure to be here today, and thanks again for the invitation to speak. So, theranostics at CHUM, clinical research integration and teamwork to improve patient access. So, me and Luke didn't speak before this, but my presentation is going to be very similar to his. I'm going to be showing how we have built the theranostics program at CHUM and what I think are our strong points and what we can improve and how we've made it work pretty well. Disclosures, so I've received honoraria and speaker fees from Novartis, Ipsen, and Spectrum Dynamics, not related to this talk. All right, so the theranostics program at CHUM in Montreal. So what's our main objective? First, it's improving patient access to theranostics. Really, that's what this is about, right? We want to get them in and get them treated. We want to improve their outcomes and their quality of life. And honestly, at CHUM in Montreal, I think we are exceptionally lucky because we have all the pieces to make this happen. It's all about putting those pieces together in a whole that makes sense and works well together. So a little bit about CHUM. So this is a picture of CHUM, downtown Montreal. So, and a little bit more info about the nuclear medicine department. So we're quite lucky. We have these brand new state-of-the-art facilities. The CHUM is located on a single site, so that's already a great start, right? Everything is happening in the same building, in the same place. Of course, there's more than one pavilion, but hey, you can't have it all. And we have one of the largest nuclear medicine departments in Canada. We have 12 full-time nuclear medicine physicians, all in one site, all at CHUM. We have 7 SPEC CTs. 6 of those are regular conventional NAI system. 1 is a digital CZT SPEC CT, a Veriton 300, the first of its kind that was installed in Canada. We have 3 PET CT. We have a cyclotron. We have 4 inpatient treatment rooms, 5 outpatient treatment rooms, dedicated RLT nursing staff, dedicated physicists. So we've got it all, basically. We're very lucky again. And all of this doesn't exist it exists in a good environment because, as most of you are probably aware, Quebec, the province of Quebec is overall lucky when it comes to nuclear medicine because of historical decisions and choices that have been made in the past. I mean, there's more PET scans in Quebec than the rest of Canada put together. That tells the whole story, right? And if you look at the number of PET scans being done, there's nearly 5 per capita. There's nearly 5 times more PET scans being done in Quebec than Ontario. So again, tells the story. And when you look at staffing, about 45% of all the nuclear medicine physicians in the country work in Quebec. So we have a better environment overall, and we have these poles of excellence in our large university center, including the CHUM, and again, that positions us very well. So when it comes to isotope production, I'm going to keep this high level. I'm not going to go deeper into one specific project, but basically we do have on-site cyclotron and radiochemistry. And our in-house production has been mostly focused on diagnostic tracers, and that's a choice, that's a strategic choice. We believe that we get more bang for our bucks when we're developing diagnostic tracers, and we mostly rely on collaboration with pharmas when it comes to therapeutic radiopharmaceuticals, right? The cyclotron and the radiochemistry lab is in the same building. It's all at CHUM. It's under the main entrance of the research center, basically. And it's all, what's very neat is there's a pneumatic system that gets the radio tracer. I don't know if I have a pointer on this. No. So the pneumatic system gets the radio tracer in about 45 seconds from the production unit to the nuclear medicine department. So they're not right beside us, but it's just like they are. One of the things that we've also decided, and I think is a strength, is we have an integrated research and clinical team in nuclear medicine. So a majority of the nuclear medicine infrastructure at CHUM is used for both clinical and research work, meaning it's more efficient that way, and we like to not duplicate either we want to avoid duplication, basically, and we like to have this integrated approach because it also facilitates the translation from the research world to the clinical application. And it's a bit of the same for the staff. We have a lot of staff that is also shared between the clinical work and the research work. And again, that facilitates the knowledge transfer, and it facilitates the move from the research to clinical, right? And of course, this is, there's downsides. I mean, it's not just all good. It means that this staff needs extra training, and they have, they can be entirely focused on either the clinical or the research, but we do believe that this is a strength and that it helps the overall program. One other thing that we have, which is great, is the Unit for Innovative Therapies. So this is a clinical core facility at CHUM and CHUM, the research center, that specializes in early phase trials. So basically, it's a whole unit. It's a 16-bed unit, both inpatient and outpatient, that specializes in early phase studies. So this is not nuclear medicine specific. This is for everyone, basically. It's for all. It's mostly oncology, and we use it. And so it regroups more than 50 investigators and more than 40 full-time staff. That's nursing staff, admin staff, lab staff. And it's this whole unit that offers basically turnkey services for phase 1 studies. So it goes from project startup to patient recruitment, to clinical service, and monitoring. It has hosted more than 70 phase 1 studies since its inception in 2019. It's the only facility of its kind in Quebec, I think maybe in Canada. And it really has allowed for the development of a very experienced phase 1 team. Because when it comes to these early phase trials, there's you guys are going to be aware of this. Of course, there's extra layers of complexity when it comes to early phase trials, particularly if you're using a radiopharmaceutical, right? And if you have all these teams all over your institution doing 1 or 2 early phase trials, they never get really comfortable with all these requirements and all this early phase stuff. So the decision was made a few years ago that we were going to have this unit that specialized in these early phase trials, and that way the staff that's working there, they do get extremely comfortable and competent at doing these early phase trials, right? And so it's really helped a lot of investigators get these studies up and running in short time and with very experienced staff. And so just I mean, we've done quite a few trials. We've been, I think, very successful thanks to all of that. So this is just a selected trial over the last few years. Everything that's bolded is a phase 1. We've had the chance to do quite a few first in human ourselves at CHUM, and we have been overall extremely lucky to be able to offer these novel therapies to our patients at CHUM. I mentioned earlier, so we do have dedicated RLT nursing staff. I think that's also critical. So the CHUM was the first center in the province to have a dedicated RLT nurse. She acts as a pivot nurse for RLT patient, basically, right? She will give clinical care, but she's also very, very involved in patient education, which is important. And she acts as, a point of contact, and that has really helped a lot, and I think a lot of center, as some of you were saying, this is going to be critical, and this is a shift in mentality, because if you go back just a few years, nobody had nurses in nuclear medicine, right? You didn't need them. You had a nuclear medicine physician and technologist, and that was about it. But now this is changing, and it will have to change if we want these theranostics program to have a good rollout. Another very important thing is training. That's also an important aspect of our theranostics program. So we are the largest nuclear medicine training, we have the largest nuclear medicine training program in Quebec at UDM. And we make a conscious effort to ensure that all of our trainees, all the residents that come through CHUM, they are not only exposed to the clinically approved theranostics, but also the research stuff, right? We want them to have as broad a view of theranostics as possible, so that if they train with us, they should be entirely autonomous to get up and running with a theranostics program once they get back to their center. We also have a newly launched Theranostics Fellowship that's starting this year, first fellow coming in a few months. And we also offer an RLT preceptorship, which is aimed at in-practice nuclear medicine physicians that are already working and they want to get up there and improve their competency when it comes to theranostics. So we offer this program. It lasts a few days. It's a mix of classroom and hands-on experience. And it's been very well received up to now by everyone that has gone through it. The other important thing is I've been speaking a lot about nuclear medicine because this is my area, of course, but this is truly a multidisciplinary approach. Most of the cases that we are going to see in theranostics, they're going to be complex. These patients, they will usually have had prior treatment, right? Because theranostics is not really, at this point in time, a first-line approach. And so most of these patients, they will be discussed in tumor board. It's extreme. We make a point to always show up, to always have someone from the team there. And communication is really key. That I think is critical. You need to have a good communication between all of the teams that are involved in the patient care if you want this to work, because it is a complex process. There's a lot of moving pieces, and because of the radioactive nature of the radiopharmaceuticals, you're on a short timetable, right? Things are decaying fast, and little mistakes can have big price tags. So communication is really key. One other aspect that I want to mention is the patient's point of view, because when we designed the theranostics program, it was critical to us at CHUM that the patient's point of view be a part of it. And so we involved patient partner in when we came up with the patient's trajectories in the hospital. So we involved them to discuss with them, what are your concerns, what's important for you. And I think that we as a community need to keep that in mind, right? We always need to get back to, okay, so what is it going to be like for the patient going through this? And so this is speaking from my experience. So most patients, truly, they are unconcerned about the exact mechanism underpinning their treatment, right? Whether it's a radiopharmaceutical or a chemical drug, they don't really care, okay? Their acceptance and tolerability of the treatment is often as important as treatment efficacy, particularly when we're in the palliative stage, which we are with most of the tracers, the radiopharmaceuticals that we're using right now in theranostics. And so treatment acceptance is really going to be dependent on when and where in the disease course the treatment is positioned, right? And what's different about theranostic? Well, it's the radioprotection, radiosafety aspect, because the treatment regimen itself is from their point of view, it's very similar to what they've had before, right? They tell us, right? It's the regimen, it's similar to chemotherapy. The side effects, well, it's nearly the same list. There's a little difference, like dry mouth with the PSMA, and okay, sure, there's some areas, but overall, where is the difference? It's the radiation safety. And when it comes to radiation safety, again, most of the patient, there's going to be exceptions, but most of the patient, they truly are unconcerned about their personal exposure. Once in a while, you'll have someone that's very concerned. My experience, again, is it's all or nothing. So either they really don't care or they care a lot and they have a lot of questions, but that's a minority of patients. And so most of them, their exposure, don't mind. But what they are concerned about is the exposure of their loved ones, their pets. We get a lot of questions about pets, the impact on their quality of life, and the radioprotection measures, particularly the isolation if they need to be alone, right? They don't like that, particularly if they're end of life, which is the case in quite a lot of patients, and the treatment regimen itself. And so to finish up, these are my thoughts. So what do we need for success? So first, we need patient acceptance and education. And here, again, nursing staff, I think, is really key, and they can facilitate the education portion a lot. We need ease of use, and that has been discussed over the last day. We need strong logistics. Availability needs to be good. Turnaround time needs to be good. And this is getting better, and I think is only going to get better as we get more of these therapies out. We need better pharmacoeconomic data. I think we've not been very good at getting those over the last few years, again, as a community, not just a SHIM thing, but we need better pharmacoeconomic data. We need to continue to foster a good, strong, multidisciplinary team approach. And lastly, I think, and I was happy to hear some of that over the last day, more combination therapy, right? Because for now, this is relatively new, and most theranostics approach are single treatment theranostics approach, but I think there's a lot of potential for combination therapy coming up in the future, and we are working on some of that at SHIM. Some of our work was presented by a student. So that is it, and I want to give a big thank you to the entire team at SHIM, because this is really a team effort, and none of this would be possible for one single person, of course. Thank you a lot. Thanks a lot for your attention.
Feeling brave and wants to come up to a mic and ask? It's a quiet crowd today. All right, I don't have a question per se, but just a comment. I had the opportunity in my past life at Novartis to visit the SHIM, and Dr. Junot was kind enough to give a tour, so really impressive new facility, having been at the old SHIM in the past. So really exciting to see what you've done there, and in particular, I will say, having been at a big pharma company where the local affiliate were always looking for ways to attract phase 1 studies from our companies, just to actually see on the flip side what you have done at the SHIM to really set yourself up for those types of trials and attract the interest.
Thank you.
So I guess just to summarize, we've come from coast to coast. We've come on this regional roadshow. We've gone from coast to coast, and from exciting bench research to bedside, or at least in a nuclear medicine department receiving a treatment. I want to thank Joseph, Kate, Luke, and Daniel for really interesting presentations. We learned sort of what's going on in BC with the innovative research in cancer. Kate talked more about applications outside of cancer care in infectious diseases and autoimmune diseases. Before shifting to sort of the clinical care with radiotheranostics, with Luke talking about the vision at UHN for a theranostics clinic. Really great approach to see where the patient literally is being put in the center, and the multidisciplinary teams and the infrastructure are being built with that premise of what's best for patients. I've personally taken that walk across the street from Princess Margaret to Toronto General's Nuclear Medicine Department and found myself wondering, how is a patient potentially with mobility issues doing this? So nice to see that there's going to be a sort of a one-stop shop approach at UHN. And then at the SHIM, where they had the opportunity as they built the super hospital to sort of think of all these things. Really nice to see that the SHIM is well set up both for groundbreaking research and for world-class clinical care. So thank you to all the speakers, and we'll pass it back to Scenic for the next agenda topic.